everyone, my name is Afonwu Neba. I am a current sophomore in the SGC program and I'm a general biology major. For my practicum, I worked under the supervision of Dr. Karen Osborne, who is in the Department of Invertebrate Zoology in the Smithsonian and MNH. So the first part of my project was mostly genetics based. I worked with the sequences of hyperids, which are marine arthropods that live in the very, very deep sea. So I would use Genius Prime to import the sequences, shorten the names. I would align them, I would trim the ends, use the correct, genetic code and check the reading frames, and sometimes translating it to amino acids can help with that. Then I would basically build the trees using FastTree. I would root them with similar species and just make sure the tree shows correct relationships. This is a great example of a great tree showing the correct relationships between hyperid families. I didn't make this one, but I did make a lot that look kind of similar to this. I also organized a bunch of spreadsheets with, for specimens, basically with their identification numbers, of course, the genus and the species where the specimen was collected, the GenBank number for the NIH side, the author, which was usually Dr. Osborne, and the voucher number. So the second part of my project was segmenting with Dragonfly, and the goal of this was to build a 3D model of a polychaete worm, and this involved highlighting, coloring in my references, filling it in, and interpolating. You kind of see me in the working space here. This is what it looked like when I was about 1/4 of the way through. And this was the final results. I did have to split my scan into 2 different ones because it became really, really big and the computer couldn't handle the massive file. As you can see, it's very defined and I left out all the hairs. You can see from another view here, you can kind of see the scales. You can see the scales here. You can see them better from this view and this view. So I learned a lot about patience and perseverance because I encountered a lot of obstacles, in both my project, and it was important that I knew to ask for help. And it also solidified my desire to want to work in research because I really admired everything that Dr. Osborne was up to in the lab and also outside of the lab. So for future scholars who are interested, I would highly recommend reaching out to her as early as possible. She is very nice and very accommodating. This is my reference for the tree photo that I referenced earlier, and all other photos were taken by me on my phone. Thank you.